Okay, let's continue our chapter 5, plasma membrane part 2. Now, a different type of transport that does not involve movement of the substance itself, but the movement of water is called osmosis. Sometimes membranes are not permeable to the substance in the solution at all. The only thing that can move across the membrane is water. Hence, in order to achieve concentration equilibrium of the substance, water will move instead to dilute one side and concentrate the other side. For example, here you have a container, okay, that is divided into two compartments by a semi-permeable membrane, meaning that it will not allow anything else through other than water, okay? These purple dots here, which represents glucose, cannot go through. The two compartments have different concentration of glucose. One side have 5% glucose, the other side have 15% glucose. This is a concentration gradient. But since glucose can't move across the membrane, what will move is water. Water will always move in the direction where there is higher concentration of substance. I used a rule: water always follow salt, meaning whatever it is going, whatever it is that is higher concentration on the other side, okay? Remember this rule about osmosis, and you can never go wrong as to which side will water move. Water always follows salt, the side with higher concentration of that substance. Now, there are two terminologies we need to define when talking about osmosis. It is solute versus solvent. Okay, solvent. So, solute is the substance, okay, that is dissolved into the solution. And in this case, they are not permeable through the membrane. That's purple dots. Those purple dots, which are the glucose, is the solute. Solvent, on the other hand, is the liquid that the solute is dissolved in, okay? And in this case is this pink liquid here, or what usually is water, okay? All right, so now before I go on to the next scenario, I want to make sure you understand the difference between diffusion and osmosis, because students always get confused on the exam, okay? So remember that diffusion, okay, is movement of the substance, which now we know we call that a solute, across a membrane, okay? And it goes according to it moves in direction of concentration gradient, okay? Gradient. So high concentration is where it will always move to an area of low concentration, okay? That's diffusion, movement of the solute itself, okay? Whereas osmosis, okay, is the movement of the solvent, or in this case, water, okay, across a membrane, okay? It's not the movement of the substance, but movement of the solvents, okay? And water will always move in the direction where there is higher concentration. of solute, okay? My water always follows salt rule, okay? So make sure you don't confuse diffusion with osmosis, okay? Because there will be problems, for example, in the worksheet that I will show you and upload to eCampus, where I'm going to ask whether this is happening, this particular substance is passing via diffusion versus osmosis, okay? Because diffusion is when we're talking about the substance is permeable toward, through the membrane, okay? Whereas osmosis is when the substance is not permeable to the membrane, and hence only water can do the movement to equilibrate the concentrations, okay? So again, another thing to add here is when solute is not permeable, permeable to the membrane. Okay, so that's the key term there. All right, so hence back to this. So there are, in the second scenario, okay, is when the membrane is permeable to both the solute, which is glucose, the purple dots, and the solvent, which is water. So now we're not talking about osmosis, okay? Because now glucose can cross the membrane. You are now in which mode of transportation again? Is it osmosis or diffusion if glucose can cross the membrane? It is diffusion because it is now the movement of solutes, which is the purple dot itself, okay, not the solvent, the pink liquid here, okay. In this case, you don't have a difference in water levels after equilibrium of the concentration is reached, because glucose will be diffusing across from an area of higher concentration to an area of lower concentration, okay? Now, so this scenario 1 is illustrating osmosis, where the membrane is not permeable to the purple balls or the glucose. This second situation here is illustrating diffusion, okay, where this red membrane here is permeable to those purple balls or the glucose, and hence glucose can travel straight across to the other side, so that you still end up with 10% glucose on both sides without any net movement of water. So that's why there is no difference in water level, whereas here we were talking about movement of water, and that's why there's this difference in water level. Okay, a related concept then to osmosis is we can classify the solvent by its tonicity, okay? The tonicity of a solution is related to its osmolarity. Osmolarity is defined by how much solute is in the solution. The lower the solute concentration, the lower the osmolarity, okay? The higher the solute concentration, the higher the osmolarity, okay? So for example, in this scenario where I have, again, a container separated or divided by a semi-permeable membrane, both sides initially have just water. But if I add a bunch of sugar crystals to one side of the container, I have just increased the osmolarity of the left side of the water, okay? In this case, because this side is now higher in osmolarity, which way will water flow due to differences in solute concentration? Now remember, water always follows salt, the side with higher amount of solutes. So water will, of course, move from the right to the left side, because now you have more solutes within this side of the container. Okay. All right, before I move on to the next slide, again, I want to draw for you the differences of different tonicity of solution. Okay, so when we talk about the tonicity of a solvent, we can divide it into 3 different types of tonicity. Okay, first one is hypotonic. Okay, when we classify one or this solvent or this side of the cell is hypotonic to the other side, it means it is lower in solute concentration. Okay, so I always like to draw my little box, okay, my little box, little tank with some solvent in it, okay, solvent, and here is my red blood cell, okay, and a little animal cell, and inside our red blood cell, we usually, in a normal basis, have about 0.9% sodium chloride. Okay, so that's why normal saline solution in the hospital, where they give you just to hydrate you with some saline solution, it is in that 0.9% sodium chloride. Okay, so if this solvent here, okay, is hypotonic, which means it is lower in solute concentration, the salt content of the solvent must be lower than what's in your red blood cell, which means, for example, it can be 0.2% sodium chloride, okay? Because this has lower solute concentration, we call this hypotonic solution. And in the case of hypotonic solution, okay, you have more solute within the red blood cell compared to the outside, okay, in this case sodium chloride. So which way will water move? It will move towards inside of the cell, okay? Remember, water will always move in the direction where there is a higher concentration, or water always follows salt, okay? Because the water will enter the red blood cell, okay, enter, all that water will enter into the cell. The cell will swell at first, okay. Remember what we talked about with plasma membrane, though. It's not a balloon, okay. It cannot expand above a certain pressure, okay, and hence it will lead to the cell bursting apart, okay, into little parts, okay, and we call this hemolysis, okay, or the cell bursting, okay. So that's for hypotonic solution. Second, we have hypertonic solution, which is now then the opposite of hypotonic, is where we have higher solute concentration, okay. So again, we have our little tank with our red blood cell in it. And of course, our red blood cell content doesn't change, still that 0.9% sodium chloride. And now it is dunked into a hypertonic solution, okay, which means it has higher solute content. For example, 2% sodium chloride, okay? So when we have more solutes on the outside compared to inside the cell, which way will water move? Towards the outside, okay, where there is lots more salt. Water always follows salt, okay. So here you have your red blood cell. Water is now escaping from interior of the cell, okay. So then it will start looking kind of like shriveled up, okay, shriveled, or what we call crenation, okay. These are terms you have to know for red blood cell, crenation or hemolysis, okay, crenation or hemolysis, all right. So that's for hypertonic hypertonic solution. And the last type is the easiest, called isotonic solution, okay, which then is when we have our little tank of solvent, dunking our red blood cell in there with 0.9%. Isotonic means same solute concentration. Okay, and hence this outside have also 0.9% sodium chloride. There is no differences in the content of sodium chloride concentration, and hence no net water movement. Okay, and then the red blood cell is happily living in its isotonic environment. Okay, so this summarizes sort of by drawing, because the way you understand osmosis is by working problems, and a lot of times the problems, when you work through them, is best understood by being able to draw these figures so you can attack whether this is a hypotonic situation, hypertonic situation, which way would water move, what happens to the cell if the water moves in this or that direction. Okay? All right. So hence now we can better understand this slide, okay, where we have our tonicity of our red blood cell. Okay, here's the red blood cell situation we were just talking about. Under hypotonic solution, the cell will initially swell until it bursts, which is called hemolysis. Okay. Or in hypertonic solution, there is a higher osmolarity inside the solvent. Okay, so water will move out of the cell. The cell shrinks in what we call crenation. And the last part is isotonic solution, where you have the same amount of salt on either side of the cell and the solvent. Same osmolarity, no net movement, and the red blood cell is happily living in that environment. Okay, now same solution in terms of the tonicity can be applied to plant cell, but the effect on the cell is a little different compared with animal cells. Remember that the plant cell, okay, has what we call, remember, what is this outer layer of the cell, plant cell? The cell wall, okay? It has the cell wall and it has a vacuole, okay? Remember, the purpose of the vacuole inside the plant is for storage of water, okay? And the cell wall is to uphold the structure of the plant cell. It's very strong, okay? So hence, when a plant is placed in hypotonic solution, it is actually a happy camper because the water is now moving into the cell and filling up the vacuole. The vacuole expands, okay, and sort of pushes against the cell wall, okay? And this situation we call the plant cell as turgid, okay, which means it is supported by a lot of power because there's a lot of water inside the plant cell, and the plant is living happily in that hypotonic solution. All right, now in the case of hypertonic solution, again, because of this existence of a cell wall, there is no problem of the cell bursting, okay? But water will still move out of the cell, including all of the vacuole, okay? And the whole cytoplasm would actually move towards the inner or central part of the cell, okay? This is called plasmolysis, okay? Plasmolysis, when the cytoplasm of the plant cell move towards the middle. And of course, if you were to dunk this plant cell back into a hypotonic solution, water will move right back in and your cell won't die, your plant won't die, okay? Now, in isotonic solution for a plant cell, okay, you have water going in, water going out, no net movement of water. And hence, in this scenario, we have not a turgid condition. We call this a flaccid condition of the plant, where it's not totally completely happy, okay, with a strong cell wall, but it's, doing all right, okay? It has enough water to survive, okay? So hence this is what this slide is illustrating in terms of what happens in animal cell being dunked in isotonic solution or hypertonic solution or hypotonic solution. Okay? So remember, in hypertonic solution, water move outwards from the red blood cell and it will crenate. Whereas in hypotonic solution, okay, you have swelling of the red blood cell until it can't go no more, and it will burst open, resulting in hemolysis. Whereas for plant cell in isotonic solution, you have a flaccid plant cell. The plant is, still surviving, is not upset, okay, or wilting, but it's all right, not completely really happy. Now, when you put the plant cell in hypertonic solution, the cell body will shrink because of plasmolysis, okay? That's when the cell wall will weaken, okay? And hence you have this wilting kind of plant. And that's why when a plant is wilting for a short amount of time, you can technically still put water in there and it will, revive, and now you have a nice healthy plant again. Okay, of course, after a couple days, it will die. Now, with hypotonic solution is where the plant is most happy in, where you have normal turgid of the cell, okay, because now water is all inside the plant cell and it's pushing against the cell wall, giving it strength. That's why it's now, sort of standing tall and upright. Okay. So in application, that's of this tonicity stuff, a doctor injects a patient with what the doctor thinks is an isotonic saline solution. The patient dies, and an autopsy reveals that many red blood cells have been destroyed. Do you think the solution the doctor injected was really isotonic? What do you think? Think for a little bit. Okay. So the answer is, of course, no, because cells have been destroyed. If cells have been destroyed, which solution did the doctor give? Right, hypotonic solution. Maybe the doctor accidentally gave it 0.2% sodium chloride instead, okay? And that's why this tonicity concept, osmosis, is very critical in health professions, okay? We will talk about it again in anatomy and physiology. And again, in the first part of anatomy and physiology and the second part of anatomy and physiology, because of how critical it is in application to the human body. You as a nurse or whatever position you are cannot give the wrong solution to a patient. It can ultimately lead to the death of the individual, okay, if you give them the wrong type. All right, so we talked about passive transport, which does not need ATP and can be divided into simple diffusion, facilitated diffusion, and osmosis. We talked about these 3 types of transport, okay? Now, these types of transport so far don't need any energy input because things are always moving according to their concentration gradient. That's what powers the movement of the solutes. However, there are transport that doesn't move things according to concentration gradient and need ATP to power the transport. We call this type of transport active transport. Before we get into active transport, though, we want to define another type of gradient that can drive solutes in or out of a cell. So far, we've been talking about concentration gradient, but there is another type of gradient called an electrical gradient, okay? Electrical gradient, which is due to differences of charges within the cell compared to outside of the cell. Generally, inside the cell right here, okay, as you can see, this is representing the cytoplasm. Inside the cell has a net negative charge, okay, whereas the outside of the cell is usually positively charged due to sodium ions, okay, being on the outside. And hence, if there are positive things floating around the outside of the cell, it will be attracted towards the inside of the cell. Let's say there's positive ions on the outside, it would want to go in there, okay? Now, sometimes both concentration gradient and electrical gradient can play a role at the same time in the direction of the transport of the solute. We call this the electrochemical gradient, okay? For example, you must be wondering why I just talked about that positive ions will go from the outside to the inside. So why is this pointing at a potassium ion to the outside? Okay, this is due to electrochemical gradient in combination. Okay, this positive charge ion, which is potassium, should be attracted to the inside of the cell because of the negative charge in the cytoplasm. But, okay, it ends up going outwards into the cytoplasm because the concentration difference of potassium ions compared to sodium ions is so great, okay, from outside and inside. The concentration of potassium ions is a lot higher, lots of purple squares compared to purple squares on the outside. So concentration gradient wins here, okay, and ends up potassium going outside of the cell. Now, in order for a lot of our cellular processes to work properly, okay, especially in the case of a neuron, we need potassium to be on the inside, not the outside, okay? Potassium ions here moving outside is a passive transport, okay, without the need of ATP, okay? Now, if we want potassium to be on the inside, this actually works against their electrochemical gradient, including your sodium ions. Hence, this is why we need the help of an active transport in form of a pump, okay? It's called active transport because it requires ATP, okay? It requires ATP to move things against the concentration gradient or electrochemical gradient. Remember, we talked about sodium potassium pump in the energy chapter, and I said we're going to see this again. So here it is again, okay? The sodium potassium pump is a perfect example of active transport, okay? And you will hear about this pump immensely again in anatomy and physiology when you get to muscles and neurons. This pump will move sodium ions outside of the cell, okay, and potassium inside back into the cell, as we have saw in that chapter 6 on energy. I showed you the video on that, okay? This is actually going against their concentration gradient. This is why they need ATP in order for this pump to work properly. Okay, and hence the application of this concept. Injecting a potassium solution into a person's blood is lethal. This is how capital punishment and euthanasia subjects die. Why do you think a potassium solution injection is lethal? The hint is, think about that normal sodium potassium concentration inside and outside of the cell is critical for signal transmission that enables heart muscles to contract. So, why do you think this is lethal? Take a few moments. So when you flood the cells with potassium, it actually messes up that whole electrochemical gradient we were talking about earlier, and hence it messes up the normal function of a muscle fiber and neuron that's supposed to stimulate movement of muscles, even those of your cardiac muscles, those muscles in your heart. And hence this is why potassium injection is lethal. All right, in cases where ATP is applied directly to transport, we call it primary active transport, okay? And the type of carrier proteins that does this type of transport can be divided into 3 categories, okay? Uniporter, which carries only one substance at a time, okay, across the plasma membrane. Symporter actually moves 2 things simultaneously across the plasma membrane in the same direction. Okay, that's why you have A going towards inside, B going towards inside as well. An antiporter, which moves 2 things in opposite direction. Okay, sort of like symporter, but it's just moving A towards the inside of the cell and B back outside the cell. And our famous sodium-potassium pump is a type of antiporter that participate in primary active transport of sodium and potassium ion. It is also called an electrogenic pump, okay, because it pumps an unequal amount of positive ions in and out of the cell. Remember from our energy chapter, we learned that sodium-potassium pump moves 3 sodium ions out of the cell for every 2 potassium ions that move into the cell. And hence, there is an imbalance of positive charge ions that are being transported by this pump, which creates a difference of net charge between the 2 sides of the membrane. Okay, this kind of gives you and reminds you of that. Remember the 3 sodium ions, okay, going outside of the cell, and then 2 potassium inside, 2 and 3. So that's why 3 positive charge stuff goes out, 2 positive charge stuff goes in. You don't have equal amount of ions moving in and out of the cell, and hence this creates a difference of net charges, okay, between the 2 sides of the membrane. If this was extracellular space of the cell, and this is the cytoplasm, now you end up with 2 different charges on either side, okay? This is how we call it. It creates a membrane potential, okay? Difference in charges between the 2 sides of the membrane, okay? All right, some transport doesn't use ATP directly, okay, but they use it indirectly. We call these secondary active transport, okay? What do we mean by using ATP indirectly? So sodium glucose co-transport is a good example. This is a type of symporter that co-transports sodium ions, okay, so these are your sodium, okay, and then you have a glucose molecule that is co-transported together at the same time in the same direction toward inside of the cell. When both sodium and glucose molecules bind to the symporter, okay, it will undergo a conformation change that flips this carrier proteins around. So now you can dump and transport all 3 of these things, 2 sodium ions and 1 glucose molecule, across toward inside of the cell. The symporter itself did not need ATP as an energy source, but it required what? Sodium ions, okay? And how do we get sodium ions outside of the cell to begin with? Okay, remember from our sodium-potassium pump, we're relying on that pump to move sodium ions toward the outside of the cell. And remember that pump does require ATP. That's what we mean by secondary active transport, using ATP indirectly, okay? If you don't understand what I'm saying and got lost, you can further visualize this with the following video clip that I will show you. Okay.
Small molecules, such as sugars and amino acids, can be transported up a concentration gradient. The sugar moves via a membrane transport protein from outside of the cell where the sugar concentration is low to the inside of the cell where the sugar concentration is high. The transport of the sugar through a coupled transport protein is driven by the movement of counterions, such as sodium ions or protons, that are moving down their concentration gradients from a region of high to low concentration.
These sodium ions.
Sodium ions and the specific sugar or amino acid simultaneously bind to the same transmembrane protein on the outside of the cell, called a symport. When the counterion is sodium, the low concentration of sodium on the inside of the cell required to transport the sugar is maintained by the sodium-potassium pump, which is powered by ATP.
Okay, so sodium ions.
In a related process called countertransport, the inward movement.
We don't need to learn about countertransport. Okay, so now we're on to the last part of this chapter, which is the last part of transport we will talk about. It is divided into endocytosis and exocytosis. Both are considered as bulk transport. Okay, so far we've been talking about movement of little substances, little molecules across either one at a time, two at a time, or three things at a time. But there are cases when we want to move a bulk amount of stuff across inside to the cell or outside of the cell. So endocytosis is a type of active transport that moves large molecules or even a whole cell into the cell itself. It does so by forming this invagination pocket, okay, right here, that will form a vesicle or a vacuole, okay. Then you will see that they're using vesicle and vacuole interchangeably. They're just both used for carrying things, transport of things, but it's just when we're talking about carrying larger particle, we call it a vacuole, okay. All right, so there are three types of endocytosis for moving this vacuole into the cell, okay? First type is called phagocytosis, okay, which is literally translate into what's called cell eating, where the particle is engulfed directly into the cell. The formation of this invagination pocket is made possible by a protein called clathrin, okay? Clathrin stabilizes this membrane so that you have this space, okay, that will stay open for it to become a vacuole. It will be eventually disengaged from this space once the vacuole is formed. Now, for transport, the vesicle and for transport, like I said, vesicle and vacuole means the same thing, okay? All right, so that vacuole will then be engulfed into the cell, okay? One example of a cell that does phagocytosis is when a white blood cell eats a foreign organism, okay? For example, if you have a bacteria that's entered the cell, like I've shown you in the previous video, okay? That shows you the process of phagocytosis, and I'll show you another video for that, okay? Second type of endocytosis is called pinocytosis, which means cell drinking. This process produces a smaller vesicle, and takes in smaller little particles or molecules, and including water that the cell needs from the extracellular space. Okay, so this is the extracellular space, okay, outside of the cell. All right, so that's why it's called cell drinking, like the cell's literally drinking. Okay, third type of endocytosis is called receptor-mediated endocytosis. This is when we want endocytosis of a specific molecule. For example, here in this picture, we have a combination or mixture of stars and squares, but we only want the stars to come inside the cell, okay? This is a targeted or specific particle to have endocytosis, okay? And so, for example, LDL receptors are necessary for moving the bad cholesterol from your blood into your, for example, other places for storage, okay? Pathogens may also, however, use this pathway to gain entry into our body cell, okay? So it can works against us as well, but in the case where in normal circumstances in a healthy individual, we want that particular specific target molecule, we want to do receptor mediated endocytosis, okay? So keyword there is targeted. All right, so this video will visually show you how the process of phagocytosis is an application of our immune system, followed by the process of exocytosis. Okay, I'm going to skip to the part where it starts talking about it correctly. Oh, let me skip to that part right here.
When bacteria or other pathogens are present in the body, certain white blood cells, categorized as phagocytes, consume the microorganisms to protect the body from infection. The process of consuming the pathogen begins when the phagocyte is attracted to chemicals from the pathogen and, through receptors, binds to it. The phagocyte ingests the microbe, encasing it in a compartment called a vacuole. Lysosomes in the phagocyte merge with the vacuole. Its enzymes then kill and digest the pathogen and release any indigestible remains from the cell. This cell process, called phagocytosis, protects the body by removing pathogens.
Okay, so, and hence that video was talking about how after you have chewed up your bacteria, you want to dump out its debris outside of the cell, and this dumping out or transporting things outside of the cell is called exocytosis, moving things out of your cells, okay? This is achieved by a vesicle moving toward the plasma membrane, just like that, or just like what's shown in this video here. It moves towards a plasma membrane, and it actually, the phospholipids of the vesicle actually fuses with the plasma membrane to dump those contents outside of the cell, okay? So the key word there is that it fuses with the plasma membrane to dump things or expel things out of our cells. So this is was one example of this happening in our bodies all the time, is how neurotransmitter is secreted from our neurons. So this is a picture of our neuron, okay? And there's lots of little neurotransmitter at the end of our neuron that needs to be released in order for signal transduction to happen. So every second of our body, okay, it is actually doing a lot of exocytosis in order for us to think correctly. Okay, so this concludes chapter 5 lecture, and I will put the practice question on eCampus for you to try out. Don't worry, it's not going to be graded, but it's more interactive that way than me reading through the question and answer choices. Okay, I will see you back in the next chapter.